Macrophages are large, phagocytic cells that eat cellular debris and other unneeded substances in an organism by engulfing and digesting them. These white blood cells are the body's first defense against bacterial pathogens that pose a threat to its health. Important organelles that control whether or not these cells are activated are called mitochondria. However, some bacteria possess genes that, when genetically expressed, get around this system by inhibiting the mitochondria's function. One specific bacteria, Legionella pneumophila, promotes mitochondrial fragmentation, which then leads to an environmental change that favors bacterial replication, causing the disease called Legionnaires. Because mitochondria are so helpful in fighting off bacteria, the bacteria have evolved ways to target these organelles when they infect the body and keep the mitochondria from performing its important duties. Previously, it was thought that the mitochondria only surrounded Legionella pneumophila bacteria that expressed the T4SS gene. However, based off of the results of this experiment, the mitochondria surround the L. pneumophila bacteria regardless of whether the gene has been encoded and expressed, which causes mitochondrial fragmentation. This breaking up of the mitochondria is the result of the effector molecule MITF, or mitochondrial fragmentation factor. The MITF molecule recruits the enzyme DNM1L to the mitochondria, which promotes mitochondrial fragmentation. This is not observed in bacteria lacking the T4SS. An important biology 1510 concept can be observed in this process. The L. pneumophila bacteria's ability to cause mitochondrial fragmentation in its host RNA translates into a protein called MITF that is released into the host cell's cytoplasm. It then recruits an enzyme to attach to the mitochondria and causes it to break apart. The breaking apart of the mitochondria creates an environment that allows the bacteria to replicate. Generally speaking, a characteristic feature of cell death is mitochondrial fragmentation, which is accompanied by the release of cytochrome C. When human macrophages are infected with L. pneumophila, mitochondrial fragmentation, which is dependent on DNM1L, still occurs. However, the release of cytochrome C is hindered, thus stopping cell signals that lead to cell death. To regulate mitochondrial respiration, the L. pneumophila undergo a two-phase process in which the first is T4SS independent and the second is T4SS dependent, while both phases are dependent on DNM1L. In the second phase, the infection causes an increase in cellular oxygen consumption that peaks at approximately 1 hour. In the second phase, the presence of T4SS yields a drastic decrease in oxygen consumption, while oxygen consumption remains high without the T4SS. Due to the vulnerability of mitochondrial respiration to the infection, glycolysis is induced. As a T4SS independent process, glycolysis is upregulated regardless of the presence of the effectors. The induction of glycolysis thus creates an optimal environment for L. pneumophila replication, creating a Warburg-like reproduction. This is understood because when HMDMs are pre-treated with 2-deoxyglucose, a known glycolysis inhibitor, replication of L. pneumophila is reduced from 46.03% replication to 11.45%. Meanwhile, the pretreatment of oligomycin, an inhibitor of F1-FO ATPase, does not have significant effects, denoting that oxidative phosphorylation and ATP production is not needed for replication. Overall, the infection of L. pneumophila creates a dynamic relationship with host mitochondria through several different, mostly T4SS independent processes. These can be observed to allow for L. pneumophila mitochondrial fragmentation, and that this fragmentation is also dependent on DNM1L. It can be seen that infected cells inhibit